All right. Good morning, everyone. I hope you had a good time yesterday at the a beautiful estate. For the ones for those of you who are in bus number 2, which included myself, I want to apologize that we took a little detour through Vermont on the way down to the. And I don't know what happened there, but luckily it was much faster on the way back. But so we're ready for day 2, and as I mentioned, since I didn't have time yesterday, I want to take a couple of minutes to tell you about the latest in lowering the input requirements. And so I think it was at this meeting, at the user group meeting last year, that I told you about the early results from our collaboration with Mar Lonisak and Matt Berriman from the Sanger Institute to try to sequence a single mosquito or a single parasitic worm, and that has since resulted in the paper that is available, and we've also written an application note and protocols. We've tested this on Drosophila as well, and so the recommendation or the protocol and workflow describes to start with about 100 nanograms of DNA, and for genome sizes up to, in our recommendation, 300 megabases. We already heard that, you know, this is, I think, conservative, because in general we have to recommend something that we believe 90% of our customers will be successful with, but that doesn't mean that at your own risk you can try to stretch that further and so forth. So for those of you who have read the paper may have seen that, you know, there were a couple of things that were a little bit not yet optimized, so I want to tell you about the optimizations that we've done since then. So one thing was about the shearing, and we heard about this yesterday. So in the paper, we described this quite faithfully, and I'm personally grateful to the editors and reviewers who let us do this. But what happened was that Mara and her team had very meticulously isolated this beautiful peak of DNA right here. And then FedEx did the shearing for us. Going to California, it arrived in just the perfect size range, and so this is why the low input protocol, as we described, didn't require any more shearing and didn't require size selection, and you go straight. And that it really allows you to get away with such low inputs because you don't have the losses associated with these steps. But clearly, FedEx is probably not going to be a life science company anytime soon, so we need to find something else, and so we've developed a low input shearing protocol using the Megaruptor 3. On the right, you just see two replicates, basically. It's 2 different mosquitoes, and starting with about 200, 240 nanograms, respectively. The yields are very good, and you can see that starting from this sort of profile where you have a high molecular weight peak and a tail, you end up with this fairly nice, and I understand that regardless of what you start out with, it'll shear down pretty reliably to this sort of size range. The second thing, and Aaron mentioned this yesterday in her talk as well, is what we call the AMPure PB size selection. So this is a dilute AMPure that you can use. Again, if you have such little material, it may be difficult to use you know, other methods like BluePippin and ELF and so forth. And that's very effective in removing up to 3, maybe up to 5 kilobase fragments. So you see here in the black trace is the original genomic DNA. And then if you do 0.45 AMPure standard, you don't really get rid of this tail here on the left side of short fragments, but in red is this AMPure PB size selection method, and you can see that that cleans it up very nicely, and so you don't have those small fragments. And so then the third thing is that for organisms like mosquito with a genome size of 250 megabases, the SQL2 capacity is too large, essentially. And so we continued our collaboration with the Sanger Institute. That's ongoing, but I want to share some preliminary results on 2 individuals, 2 mosquitoes now that are barcoded and underwent this, what I just told you about, the low input shear and this AMPure PB size selection, and you get this red peak of about 15 kb or so. And then the sequencing results, 1 SQL2 smart cell, almost 300 gigabases raw yield, 200 kilobase read length, and 50, and the insert N50 is 12.5 kilobases. And so because we're choosing to shear down to a size where you do get multiple passes, you have very good barcode recall yield, so 83% of the reads had a barcode, and actually 98% of the bases, because the non-barcoded reads were very short and likely early terminations, which is the reason why they didn't get the barcode. And so then, again, these are just 2 different Colizia mosquitoes, 2212 and 2213. They assemble very well. The right genome size, the 1 megabase Contig N50 club, BUSCO, very complete and so forth. But I told you that the peak is around 13 kb, so that actually means that we're going to get quite a lot of HiFi reads. So we were interested in how the two data types and assemblies would compare from the same data set, looking at CLR assemblies versus HiFi assemblies. And so that's shown here. So for the one sample now, 2213, we're going to take the same data and do the CLR assembly that I just showed you and compare it to the HiFi-based assembly that I talked about yesterday. And you can see that it's actually better. So the HiFi assembly has a higher contiguity, still the same completeness, and significantly it required a lot less compute time. Now, to be fair, it did require some time also to calculate the CCS, the sum of which is still less than the CLR assembly. But as I told you, this bottom line is going to go away, and so there's going to be like a factor of 5 or so less compute time. And so then the other thing that you notice, and that I'm going to close with, is that the HiFi assembly is actually 10% larger than the CLR assembly, and about 200 more contigs. So we were interested in what are those extra contigs. And so they are real mosquito sequence, and so they tend to be highly repetitive, these satellite sequences. Many of those map to what's called the pest unknown. So the pest is the mosquito reference sequence, and it has a bucket. Oopsie, sorry about that. It has a bucket of unassigned and unplaced contigs, and those are mostly represented here, and of course the HiFi contigs are much longer than these unknown contigs. Unknown contigs tend to be like 1 or 2 kb, and here on the right I show an example of 137 kb contig that is specifically only in the HiFi assembly and is missing in the CLR assembly. And if you map that, you find that it's 41% made of this AGX 367 satellite sequence, which is 367 base pairs. And so that was known, but what was not known is how that's arranged in the self-dot plot. If you're familiar with these dot plots, whenever you see a knitting pattern, that means it's highly repetitive sequence. And so we're excited about that because it's yet another example of the HiFi data getting through these. So remember, this was a 13 kb library, and we're resolving 137 kb of this highly repetitive sequence, and that was collapsed in the CLR assembly. So another example of our belief that the HiFi approach gives you more complete genomes. So the low input protocol is now available. We have the procedure and protocols. There was a webinar last week that has been recorded, which if you want to hear more about iceworm, beyond what Aaron talked about. And so we are excited about this because I think it's going to likely enable many other applications. And then looking into the future, we are working on lowering that even further. So this is currently in development, and we don't yet have a time frame for this, but I want to share with you some of the initial work and our intentions to drop this down to even lower, about 1 to 5 nanograms, so that you can get even at the very small critters, worms, and maybe even tardigrades and so forth. We do believe that it has other application spaces for like cancer needle biopsies, where you get a few thousand cells, or scraping a colony off a plate without then having to wait the overnight culture of bacterial and so forth. So we are testing this now with, this is a test sample, yeast, but we are very pleased initially to see that from 5 nanograms of input DNA, we got 500 gigabases from a single SMRT Cell 8M, very good performance, yield, reads, and so forth, and the HiFi stats were very good, 2.8 million Q20 plus reads, 1.1 million Q40 plus reads, so they're basically error-free, and average insert size. And so now you can do an assembly. Now yeast, we have tons of coverage, but just to show you that there was really good coverage and not a lot of bias and so forth. And then we did an assembly, which was, so the reference is the theoretical that you can get, because yeast 16 chromosomes and the mitochondria, and the Contig N50 is what it is because the chromosomes have a fixed length, and we're quite happy with the initial results. So this is in development, and so stay tuned, but I wanted to tell you that we're certainly not done with lowering the input, and we think there's going to be good things to come in the future.